All right, good to hear. So you guys have 4 of those, and they're going to be a lot of fun, especially the cranial nerves. I remember having a lot of fun with that one. So this week actually, before we get started, I would like to just bring up a question that I got sent an email about. Yeah, so if I get good questions sometimes, guys, I'll just bring them up with all of you, just so it doesn't because some of you might get that question later on as well. So this is what we did last week. We looked at the ventral surface of the brain. At number 12, you can see the hypothalamus is indicated. And the question was just that the student had seen this sometimes labeled as pituitary gland. And you will see that occasionally. I've seen it in plenty of diagrams. So it's just, there's a really small dot, I don't know if you can see on the screen, a really small dot underneath the hypothalamus, which is your pituitary gland. So it kind of hangs off the bottom of your hypothalamus. So sometimes if you see just like a line drawing diagram like this one, that's just, yeah, sometimes it'll be labeled hypothalamus, sometimes it's pituitary. It's not really have enough fine detail in the diagram for you to be able to know either way. So we usually wouldn't like expect you guys to know the difference between that. But if in your during your anatomy stuff with your anatomy tutors, if you ever get the chance, maybe ask them to point out the difference between them, because it might be something you want to know. But yeah, that's all I want to say about that. This week we're doing synaptic transition. So I apologize if too much of this seems like revision, because I know you had your lecture with David already. But otherwise, yeah, we're gonna get straight into it. All right, cool. Okay, so just a bit of summary, revision first. So the human nervous system communicates using a vast network of interconnected neurons. So neurons have these really long bodies called axons, which is an important way that they function. And these long bodies will connect with other neurons at these gaps that we call synapses. So we use electrical signals called action potentials to drive signals down the body of a neuron. But when that signal needs to transmit to another neuron, they use neurotransmitters. So from one length of a neuron to the other, it's an electrical signal. And then when that signal needs to jump to another neuron, it is a chemical signal. And then when it sort of passes the synapse and it goes to another neuron, then it can turn back into an electrical signal. So a little bit more on that later. This is just sort of general summary. And those chemical messengers that we use are called neurotransmitters, which obviously we will be learning a lot about in this subject. Cool. So the all or nothing principle, pretty sure David would have mentioned this a little bit during his lecture last week. So again, just to summarize, an action potential is when the electrical potential of an axon reaches a threshold value. For neurons, this is usually a set limit, but it can differ between different types of cells. So like cardiac cells might have a different threshold compared to neurons. If the stimulus greatly exceeds the threshold, the action potential will still be the same size or magnitude. So that is kind of an important landmark feature of action potentials. And if the stimulus does not reach the threshold, like it's too low, there will be no action potential. And that is just the all or nothing principle. So as the action potential travels along the length of the axon, it triggers the next section of axon to fire, and the magnitude does not change as it travels down the axon. So generally, the magnitude of the action potential stays the same. I would, yeah, just be very aware of that. That's one of the core features of action potentials. The way that sort of magnitude works with action potentials, if you want to phrase it like that, is all about firing frequency. So neurons that are able to fire more quickly, that is indicating a stronger stimulus. It's not the actual electrical current that is sort of becoming like more powerful. It's more to do with frequency, or how rapidly the neuron is being activated. Okay. So this diagram is from your lab manual, and I guess the activity that we used to do when we were in class was just to get you guys to fill this out. If you haven't already had a chance to do so, and some of you might find it really helpful. I know, understandably, if a lot of you are sick of this by now, because you've pretty much done it every semester so far throughout medical science, and those of you who aren't doing medical science, you do need to know it for this subject. It's always helpful to do a little bit of revision. So all you have to do for this diagram is just label when your sodium channels like open and when it enters the cell, when they close, and then when like potassium channels open and they let potassium out of the cell. And you also need to be able to label the depolarization and repolarization phases and your refractory periods. That is pretty important. I would definitely make sure that if you guys aren't super familiar with this already, just re-familiarize yourself with it now. But it's not necessary for you to have to draw this all out again if you don't want to or don't need to. This is explaining, yeah, good. So these are the refractory periods. So during depolarization and repolarization, the membrane is unable to undergo a second action potential, so it must recover. And this recovery period lasts for about 1 millisecond, and it is known as the absolute refractory period. So between the start of the stimulus is or start of the depolarization, that's your absolute refractory period start, and it ends about after 1 millisecond after that. Around the time when your sodium channels close. So during that full depolarization period, that is your absolute refractory period. And no matter what happens, you can't have another action potential generated during that period. Pretty straightforward. So to recover, the second inhibitory gate must open, which resets the channel. And after this is your relative refractory period. So this is can last for a couple of seconds. It lasts pretty much up until the end of your hyperpolarization period, when your cell returns to a resting membrane potential. And during this time, you can have a second action potential generated, but it must be a really big stimulus. Usually it needs to be a lot bigger or more intense than the original stimulus that produced the first depolarization. Okay, so this, you also have, after this we would talk a little bit about like just some examples of how potassium channels work and how like gradients work. So there's a couple of questions from the manual, we'll just go through them now. So hypothetically, you have prepared some potassium leak channels. Predict how the potassium ions will move under the following conditions and what sort of membrane potential will develop. And as a note, potassium channel works regardless of the orientation. Yes, I was just saying that the way this question is worded, just remember that potassium channels work the same way regardless of which way they're facing. So potassium will always move from a point of high concentration gradient to a point of low. So your first one is, where will potassium ions move if you have equal concentrations of potassium inside the cell and outside the cell? What do you guys think? Will it move? Will it not move? It will not. Yeah, you'd be correct. There won't be any movement. So if you have equal concentrations inside and outside the cell, there is no concentration gradient. So you won't, you won't you won't have any movement of ions. So the second one, if you have only potassium ions present inside the cell, you will have a net movement of potassium ions to the outside. Again, just kind of simple concentration gradient stuff. It's really just it's still just revision at this point. Try not to spend too much time on it. The membrane potential with regards to the inside of the vesicle will become more positive until the concentration gradient no longer exists. That's just physics. That's just kind of nature. And then obviously, as potassium ions are present only on the outside of the vesicle or the cell, this will cause a net movement of potassium ions to move inside the vesicle. Cool. Then we have a question regarding transmedicated ion channels open in response to specific neurotransmitters in the environment, but are insensitive to the membrane potential. Therefore, they cannot by themselves generate an action potential. So you guys probably would have known a little bit about ligands and chemical messages and this kind of thing. This is just a simple true or false question. Do you believe that this statement is true or false? What do you guys think? It is true, yeah. So the activation of the ligand receptor is required to open the ion channel in which it is coupled. So some ion channels are just specifically, they need to have their specific ligand attached to them. And without that, they're non-functional. So some of them are completely irresponsive to action potentials, and they just require their sort of specific ligand to attach to them. Just a different way that some channels function. Okay, then we have like 20 multiple choice questions that we're basically going to go through today. Even more, yeah, we have like 30. Great. So a couple of different ways that you can, I guess, follow along with this. You guys might want to be taking like small notes. You might find it most helpful to just write down the whole question. Sometimes we do use questions in our exams that are similar to ones that are like presented in the tutorial. I don't think we would necessarily just like copy and paste questions from like the lab manual, but yeah, it's important to know. Like all these questions we can potentially bring up again. So yeah, they are important. Okay, so what is the principal way that signals are transmitted between neurons in the adult human nervous system? You guys might actually know some of these. Do we think it's A, B, C, D, or E? And if we're just sort of looking through these, there's one of them that probably stands out most, just in terms of, I guess, what is most relevant. D. It's a good answer. It's a good answer. Chemicals are released into the synaptic cleft and bind to their specific receptors on the postsynaptic membrane. Yeah, it's exactly right. It's the most correct here. The rest of these are sort of talking about things that aren't really relevant. And we're actually going to get a little bit more into this now. So you have, yeah, I don't have the original diagram, but what we would then get students to do is get them to draw a line drawing of a synapse. So I have done one myself. Obviously, I'm not a great artist, but it's pretty good, and this is about as good as I would expect you guys to be able to do. I encourage you guys to have a go at this. So the full diagram is going to look something like this, and I'm just going to walk you through it now. If you want, grab a pen or a pencil or whatever, and you can follow along with me and just be writing down like and labeling this diagram as we go along. But this is like how a synapse generally looks. And you will find it very helpful if you write all this down. You should find it pretty helpful because it's going to help you answer some questions. And as you remember, your final exam is going to be open book. So having like a really simple diagram like this is a really quick way to just like, you know, you can flick back through your notes, see this diagram that you've written, and it will actually help you answer quite a few different questions. Okay. So this little bulb thing at the top left is your presynaptic terminal, also known as just your axon terminal. So an action potential will travel down the neuron, and this is where it ends, basically. Then you have your postsynaptic axon. That's where our signal is going to end up. And the gap in between that is called your synaptic cleft. So we have your presynaptic membrane in red, and you also have your postsynaptic membrane in red. That should make perfect sense. You've got presynaptic membrane on the presynaptic axon and so on. And on your presynaptic membrane is where you have your active zones. So these are where, it's really important that you remember this. I have seen this question pop up before. It is where your vesicles attach to the membrane and release neurotransmitters, which I'm about to show you. So your vesicles are what contain your neurotransmitters, and when they are told to, they release neurotransmitters into the synaptic cleft. And they do so by binding to the active zone on the presynaptic membrane. So just to kind of go from start to finish of how this all works, I've kind of done like a step-by-step process of how this might work. So one is where the action potential travels down the axon and arrives at the presynaptic axon terminal. Here it causes calcium release. So you can see a little calcium over here. And calcium is what causes your neurotransmitters to be released from their vesicles, or it's what causes your vesicles to bind to the active zone and then release their neurotransmitters inside. So your vesicles are release their neurotransmitters into the synaptic cleft. They then sort of flood the cleft, and they really don't have anywhere to go except land on the postsynaptic membrane. And here they bind to specific receptors that are looking for them, and that neurotransmitter binding causes that depolarization to continue along the next neuron. So it's basically continuing the action potential. I don't know, it seems a little bit convoluted, going from an electrical signal to a chemical signal to an electrical signal. It's kind of deeply rooted in evolution, why we like do all this. But certainly when we look at like diffuse modulatory systems in a couple of weeks, might be the week after next, I think, it will start to make a bit more sense why we use chemical messages as opposed to just having everything be like electrical based. Neurotransmitters are incredibly powerful. They have a range of uses, and importantly, they have different uses in the central nervous system compared to the rest of the body, which is pretty interesting. So here's your full diagram of how a synapse works, basically. And there's a whole bunch of questions that, that's evolution is really difficult to reason at points. There's a whole bunch of different reasons that adaptations came about, and for the most part, a lot of it is guesswork. So yeah, so for instance, like this calcium thing is a question I see all the time. So calcium is required for neurotransmitter release. It is not directly an action potential that causes neurotransmitter release. It's calcium. Calcium in itself, though, is released by an action potential. So it's more about just remembering that sequence of events is really important. I think that's the main takeaway from this diagram. It's the main reason I would encourage you to physically draw this out. And other things, just like remembering where active zones are, I think can be an important thing to remember. All right, moving on. More multiple choice questions. Which statement is the most correct about neurotransmitters? Give you guys a little bit of a think. Even if you don't want to write it in chat, just have a think about it, have a read. In your mind, just kind of be like, yeah, I think it's C, or I think it's B. It's certainly not A, but it is B. So stores of neurotransmitter are located in the axonal terminal. And at the very end, we will talk about how, yeah, a little bit more about how like they're stored and how they're produced. This is like really broad because neurotransmitters, there's 4 main ones that we look at in this subject, and right now we're just talking about them very broadly. So just bear that in mind. Question 4, which statement is the most correct about neurotransmitter release? So a couple of things here which are looking to sort of trip you up. And this is what I was talking about before. because the answer is E, where just having that diagram might be a helpful way of remembering the sequence of events. So localized increases in calcium are what induce neurotransmitter release. Literally what we were just talking about. And the rest of these, yeah, no, not relevant to what we're talking about. It's not going to cause neurotransmitter release. Moving on, question 5, how do neurotransmitters exert their effect on the target cell? Should be reading A and thinking, seems pretty reasonable. Don't see too much wrong there. By binding to specific proteins located in the plasma membrane of the target cell. Yep, it's all about that specificity. You guys would remember the like lock and key model of how receptors and proteins and that kind of thing work. It's the same thing. Specific proteins that are looking to interact with specific receptors. And answers like C and D, like forming sodium or chloride channels, that's more so what like hormones would do, where they physically like enter the cell, their target cell, and cause them to build like more sodium or chlorine channels. Not really what neurotransmitters do, no. Question 6, what is the principal process involved in terminating the signal at the synapse? I'm not sure if you guys went through this with David, but it is where concentrations of the neurotransmitter get reduced. Typically, it is through enzymes that specifically degrade the neurotransmitter. So for each neurotransmitter or sort of like class of neurotransmitters, there will be specific enzymes that break it down once it's in the synapse. And that just stops you from, you know, for example, having like serotonin or dopamine just sitting in the synaptic cleft and just continuously interacting with your postsynaptic membrane receptors. And that is something that we come back to when we do SSRIs and stuff like that. So selective serotonin reuptake inhibitors, which is a really long word for basically a class of drugs which stop serotonin from being degraded by enzymes in the synaptic cleft. So definitely a topic that we'll be revisiting. Question 7, for which of the following neurotransmitters is degradation in the synaptic cleft the principal means by which its effects are terminated? And it is acetylcholine. So not all of them are primarily relying on being degraded. There are other ways in which their effects are terminated. But in this case, it is certainly acetylcholine. Question 8, which of the following neurotransmitters are synthesized from amino acids? So it's actually A and B, serotonin and noradrenaline. Glycine is itself an amino acid, so it doesn't need to be synthesized from amino acids. But glycine is itself also a neurotransmitter. Not really one that we look at. It's not really one that we look at a whole lot in the subject, but serotonin and noradrenaline, certainly you'll be like hearing about those all the time. Although we're going to talk about it right now, apparently. So when glycine binds to its postsynaptic ligand-gated receptor, which of the following will occur in the postsynaptic cell? And it's going to be D. So this is what normally happens with inhibitory neurotransmitters. They increase the membrane permeability to chlorine. Chlorine is sort of thought of as having inhibitory properties. One thing to think of when you, or one thing to associate with chlorine, I guess. Moving on, activation of the nicotinic acetylcholine receptor means that postsynaptic neuron is, which of the following? So have a think about nicotine. Have a think about whether it is stimulatory or inhibitory. And I guess another way of thinking about this is think of nicotine in cigarettes. So cigarettes cause like an increase in heart rate, can cause increase in blood pressure because nicotine is a stimulant. So activation of nicotine receptors is stimulatory, yeah? So we'll reason that far. Maybe keep that in the back of the head while you're reading these answers. So activating nicotine receptors is going to cause the postsynaptic neuron to be more likely to be responsive to the generation of an action potential. So that is, in essence, what stimulatory neurotransmitters do, or stimulatory receptors do. They lower the, I guess, threshold required for an action potential, and so that's what makes it more responsive. So lower threshold means that the resting memory potential doesn't have to come up very high. You don't need a very strong stimulus in order to get that initial meet that all or nothing principle, and to get your full action potential or your full, I guess, response. Question 11, which of the following are not examples of transmitter-gated channels? So all of these are ionotropic receptors except for C. Moving on, which of the following is not correct about NMDA-gated channels? It is C. Agonists of this receptor are used recreationally for their hallucinogenic effects. That's not true. I believe NMDA-gated channel drugs, I think they're usually used for like pain. They're usually targets for pain relief. And agonists, guys, you know what agonists are. If any of you are pharmacology students. So agonists means to like activate or exacerbate, and antagonists are just the opposite. It means to like block. Question 13, what is the role of G proteins in G protein coupled receptors? So G proteins are really important. It's where you have one tiny molecule or receptor. So when the molecule binds to that G protein coupled receptor, it causes that cascade effect. So one molecule has, through a cascade effect, a really big impact. Like it can through this idea of cascade signaling, can have a really big impact. So the answer is B, activation or inhibition of effector proteins in response to receptor activation. Question 14, which of the following statements are correct? So is it that metabotropic receptors act as channels for low NW molecules that cannot traverse the plasma membrane by simple diffusion? Don't think so. Ligand binding to metabotropic receptor does not affect postsynaptic memory potential. Also don't think so. Or is it that metabotropic receptors can recruit second messenger pathways to mediate the signal of ligand binding? Sounds great. That's the whole idea of second messenger pathways. And again, it's through the cascade signaling, or the cascade pathways. The idea, it's like where one pathway activates 2, which activates like 4 pathways, which activates 8, and then it's kind of got this cascade or like waterfall effect of signaling different molecules and just recruiting lots of molecules to enact a specific function. All right, question 15. Which of the following events can occur in response to a ligand binding to its metabotropic receptor? So protein phosphorylation, changes in potassium permeability, or increase in potassium permeability, and phospholipid or fatty acid metabolism. It is actually all of those. All of those can be caused by the activation of a metabotropic receptor. Special proteins that form channels allowing ions to pass from the cytoplasm of one cell to the cytoplasm of the next are what? And they are actually connexions. So these are just what allow, they're kind of like backdoors that allow neurons to share some neurotransmitters as well as some signaling. Some signaling. 17, electrical synapses are so fast that an action potential in the presynaptic neuron can instantly produce an action potential in the postsynaptic neuron. That is true. We haven't been talking about electrical synapses, we've been talking about chemical synapses, however. They're not instantaneous. They are very quick though. I know it seems like a slow process where you're flooding a gap with, or a synaptic cleft with chemicals, and you're just waiting for them to swim across to the other side and activate receptors. I know that might sound like a slow process, but it is incredibly quick. Because we're talking about neurons, and we're talking about the same pathways that cause you to have incredibly quick like reflexes and that kind of thing. So they are still very quick pathways. 18, active zones are located on the presynaptic side. Yep, that's exactly what we were just looking at before. 19, which of the following is an example of a receptor agonist? So something which exacerbates or activates a receptor, and it is nicotine. So nicotine acts on nicotinic receptors. And these top two are actually central nervous system depressants. So their action, I believe, is mainly antagonistic. And I believe curare causes muscle paralysis, and it's used in like poison darts. This is pretty cool. Question 20, how many transmitter-gated channels are activated during synaptic transmission is dependent on the amount of neurotransmitter is released. That is released, yes, that's absolutely true. And that's one way where if you have like all your receptors are already taken up by their specific ligands, then you cannot increase how much of that is being used up. So there is like a maximal amount that can be put in. 21. Drugs that bind to receptors and block the normal action of the transmitter are called what? They're called antagonists. 22, in situ hybridization, which just means in lab, can confirm that a cell synthesizes a particular protein or peptide. That is true. I haven't really seen this question come up, but it's something that you should probably just remember it. Just remember it. It's a weird one, but it's worth remembering. Question 23, AMPA receptors are subtypes of what? They are subtypes of glutamate. So you have NMDA was one that we saw before. That's also a subtype of a glutamate receptor. AMPA is one of them, and oh yeah, so the top one there, kainate, is the last one. So NMDA, AMPA, and kainate. 24, cholinergic and non-cholinergic neurons can manufacture acetylcholinesterase, which is the enzyme which breaks down acetylcholine. That is true. So the neurons that use acetylcholine can manufacture the enzyme which breaks down acetylcholine. That's all that's really asking you to understand. 25, when catecholamines are released at a high rate, the activity of TH increases. 26, catecholamine inside the axon terminal may be reloaded into synaptic vesicles and reused, or they can be enzymatically destroyed by the action of monoamine oxidase. That is true. So some of your neurotransmitters will specifically be acted upon by monoamine oxidase. And I don't know if you guys have heard of monoamine oxidase inhibitors, sometimes just called MAOIs. But they are similar to the idea of SSRIs in that they are a group of drugs that target the reuptake of neurotransmitters. So it's basically trying to keep them in the cleft for longer, working more. Certainly in the cases where for that specific neurotransmitter, their main way that they stop working is that they get degraded by these enzymes. So if you stop those enzymes, they're just gonna work longer, basically. That's what makes these classes of drugs like SSRIs and MAOIs so good at targeting like depression and anxiety, and certainly symptoms of those. But we're obviously coming back to that. Have a whole tutorial just on anxiety and depression. 27, serotonin reuptake may be inhibited by fluoxetine. Yeah, won't go too much into that because, again, we've got a whole tutorial on it. 28, which of the following glutamate-gated ion channels causes a large and rapid depolarization? It is AMPA. NMDA is, causes a much slower and smaller depolarization. 29, signal amplification can occur at several places in the cascade, but only one ion channel can be activated by a G protein. That is true. And let's move on to the last one. So what are the 4 basic requirements for using chemicals as messages in the nervous system? So leave a bit of space to write this one out because you need to know all 4 of these dot points. So the first, I guess, requirement is that you must be able to synthesize that neurotransmitter in the presynaptic neuron. So at the place where it's about to be released. You have to have a space to be able to store it. So in this case, it's your vesicles, broadly speaking, for neurotransmitters. The third one is that they need to be released from the presynaptic axon terminal when stimulated. So broadly speaking, as we mentioned, you have when an action potential causes calcium release, and calcium, or the buildup of calcium concentration, is what causes your vesicles to release neurotransmitters. And finally, when those neurotransmitters act on their target receptors in the postsynaptic neuron, they need to produce a response, whether that response be like a new action potential or whatnot. So those are your 4 basic requirements. I have definitely seen that as a past question. Definitely know and be familiar with that. And then we can go on to looking at a specific example of that, just so you guys are a bit more familiar with this. And just to really drive home the idea that, yeah, this is like a really important part of the tutorial. Yeah, so we're going to use the example of acetylcholine. So synthesizing acetylcholine in the presynaptic neuron. How do we do it? We take choline acetyltransferase, which is an enzyme which makes acetylcholine out of choline and the acetyl group from acetyl-CoA. Yeah, and you can see that at number 1. So CHAT is taking choline and just the acetyl group from acetyl-CoA, and it's turning that into acetylcholine. Number 2 is that acetylcholine must be stored in the presynaptic neuron. It does so in vesicles. Number 3, it needs to be released from the presynaptic axon terminal when stimulated. Yep, that does so. And number 4 is it needs to produce a response in the postsynaptic cell. And acetylcholine does so at muscarinic and nicotinic receptors. So that's just an example of acetylcholine. As revision, yes, more homework, sorry. If you want it, you can use the example of norepinephrine or dopamine if you want, just as extra stuff for you, just because I haven't given you enough work already. And these kind of diagrams, if you can find ones that are similar to for dopamine or noradrenaline or whatnot, these kind of diagrams are really good. They just really show like a step-by-step process. I've added in these numbers 1, 2, 3, 4 just to illustrate it. But this is generally how your neurotransmitters work. All right, that went quickly. I can't believe it was already 3:45. So before I let you guys go, Taryn mentioned in an email, sorry, not an email, in an announcement that she was hosting the DMP questions and answers. So that is going to be at 4 o'clock. That is going to be in 15 minutes. If I can, I will just post the link now in chat. Zoom link. There it is. So it's starting at 4 o'clock. You don't have to go to it if you haven't thought or like just put any thought so far into the DMP. I recommend just going and just seeing where everyone's at, because you will find that people are asking questions that are important at this stage in your digital media presentation. So even if you don't have questions yourself, I encourage you to go. You might think of a question when you get there. You might be like, oh, I just realized that Luke and Taryn didn't cover this in that first week, so this is definitely something that I want to know before I get started. Other than that, yeah, Taryn has mentioned, as well as I have, that the best place to start is really just like starting with a script. I'll let her get more into that, but it's basically just like start doing research now, start getting some sources together, finding some really good websites in terms of like getting information. Be sure to write down all your like references as you go along, so you don't have to just like make up or like find a bunch of random references at the end and pretend that you use them. Just do it as you go along. And yeah, start working on that script, which is it's just basically like start with a lot of research, and then from that point on, you can move to like making it more like a little bit more informal, a little bit more casual, because we want to feel engaged in that kind of thing. But I'm going to let Taryn sort of cover all this for you guys. Hi Luke, will answers be posted for last week's homework? We usually don't. I can talk to Taryn about that, and I'll probably have an answer for you for next week. So the last week's revision was, there was some like questions on like the sympathetic and parasympathetic division and where they like originate from your nerves. I think there was like 5 questions. So I'll talk to Taryn about it. I'll have an answer for you guys next week when you're back here. And next week we are doing EEG, which is very cool topic. And I'll get to show you guys like a video demonstration of how to attach an EEG that we filmed at UTS earlier this semester. But yeah, thanks for coming, guys. Go take a quick break, 10 minutes, and then it'd be great if as many as possible of you showed up to Taryn's Zoom call in 10 minutes. I will also probably be dropping by, but I won't be doing anything. I'll just kind of be lurking like most of you guys do. Thanks for coming, guys. See you next week. Take care. See you guys.
Bye.
Bye.
Thank you.
Thanks, mate. See ya.
See you guys.